grafting session a little later on this afternoon. Please let me introduce Dr. Hassell from Clemson University.
Thank you. We're going to talk about grafting. So I've been doing grafting for a number of years now, and I'm far enough away from home, so I'm considered an expert. I think you have to be 40 miles away, so that puts me as an expert status. But we're going to have a lot of fun with grafting. If you have any questions as I go through this and something you don't understand, just raise your hand, interrupt me. I want to have more of a dialogue with you as we talk about grafting, because it's so very important that you understand the mechanics of grafting. Grafting is both an art and a science. And so to become a real successful grafter takes time and practice. And so it's not something that, that you can pick up readily and do. I've had failures, I've had a lot of successes, but you have failures, and you try to figure out what's going on and what's happening, and so it's a learning process as we go along. But the title is Why Graft? Why do you want to graft? You know, and basically if you're looking at watermelons, you want to graft because watermelons generally have a terrible root system. Watermelon root systems are by far one of the worst root systems of all vegetables. Okay, right up front. It's a terrible root system. If you go out there and dig up your root system on a regular watermelon, you'll wonder how it ever produced a watermelon to begin with. Okay, it's really poor. Okay, the breeders have never paid any attention over the years. They're always interested in the final outcome is what we eat. They're interested in flavor, size, sugars. Nobody's ever really paid attention to the root system. And so grafting has come about because of poor root system and of soil-borne diseases. And that's the biggest problem. This just shows you, this is one of my favorite pictures. This just shows you a watermelon on top, but this happens to be a gourd rootstock. This is a bottle gourd rootstock here. And you have to remember that when you're grafting, this is no longer, this top part is a watermelon, but below ground, it's not a below ground watermelon anymore. It's a below ground gourd. Okay, and how you handle that, and how you grow it, is very different than growing a watermelon. Okay, so the root system is entirely different. Now, the reason you graft, there are several things you want to look at. And for you, you're looking at fusarium wilt resistance. That's the big number one. Okay, fusarium wilt is in the U.S., and as far as the world goes, it's a soil-borne problem. You have it in your soils. Rotation doesn't work real well. Once you get fusarium in your soils, it's done. Okay? And there's a lot of theories out there that say, well, you can apply this, you do this rotation, and you'll get a little bit of effect, but generally there's really no cure. Nematodes, that's the second worst problem. Okay, it looks like you have, if you have nematode problems, you recognize that real quick because it looks like huge clubs all over the place in your root system. And these two right here are basically interchangeable. If you have this one in our soils, you end up with this one. Phytophthora, big problem. Sudden wilt, big problem. Okay, but also the main target, you're looking at improving adaptability to cold, to heat, to wet, to salt tolerance, salt problems you have in your soil. On the coast now, out west, in our coast, depends on the year. You know, every year we end up with different weather forecasts, and you know, at the end of the year, it all say, oh, it's a normal year. And yet, you had a terrible wet spring, or you had a real dry year, but then all of a sudden the skies opened up and you got sufficient water. It was a normal year, right? You always get those reports. Normal year. But we have a lot of salt problems. A lot of our wells on the coast now are getting salt intrusion, so we're actually irrigating. A lot of our wells are now have salt water, so we're irrigating. We're more or less irrigating with salt water. So we want a root system that can tolerate that. And then these are just increasing our root system on grafting. It'll impact our fruit size, our shape, our sugar content, and our texture. Most important with grafting, though, it extends our harvest period. Because the plants continually grow with grafting. Okay, so that's a big advantage, is the plants don't go down. Okay, and so it's one of the biggest problems we have with grafting is grafted plants have a hard time switching from vegetative growth to reproductive growth. Okay, that because the root systems are so vigorous that they want to stay more vegetative. Okay, so that's a big problem we have in commercial production, which should not be a problem for you, but it is problems for watermelon growers, and they're interested in that first set. They want to make sure that they get on the market early. And with grafting, that's the real problems we're having with that. This just gives you an idea what history of grafting goes. We're in the U.S. here, we're all new to this. But it's been going on over in Asia for many years. And I'll show you, I mean, as far as grafting goes, we've been behind the 8 ball for a long time on grafting because we haven't had the need. We've always had land to rotate. We've always had soil fumigation. So to combat those soil-borne diseases, so we really haven't paid it much attention until probably the last 10, 15 years. This is what I wanted to spend some time with you on, because this is extremely important that you understand. If you're going to go watermelons, this is the rootstock here is the lagenaria. That's a bottle gourd. Okay? And if you're going to grow anything else other than watermelons, you cannot use a lagenaria. Okay? Lagenaria only works for watermelons. And there's only certain species of lagenaria that work the best. And we're going to talk about some incompatibility issues in a little bit. But just because it says it's a bottle gourd doesn't mean that it's going to work for you. Okay? They've selected certain ones over the years that have worked the best for grafting. And it all comes from Asia. Okay? All these rootstocks are originated from them, and they've selected them over the years. So you need to understand that just because it says it's a bottle gourd doesn't mean that it's going to work for you. Okay? That's extremely important that you understand that. Most of the world uses what they call the interspecific hybrid rootstocks, the squash rootstock. They're crosses between a Maxima squash and a Marshata. Okay, and this is a cross that's used for watermelons, melons, ornamental melons, cucumbers. It's the world one that's accepted all over. And there are certain ones that they've bred over the years that work to enhance, has the resistance, has the tolerance to the diseases that you're after, but it also increases the production. Okay, and they are compatible. These same things, the Marshatas works real good for the cucumbers. The Maxima works good for the melons and ornamental melons. The wild watermelon, the Linnaeus that you have, works only for watermelons. The Cucumis mellow only works for melons. And then this one, I've never ever got a hold of any of the seed, but this works exclusively for cucumbers. So even though it's a cucurbit, not all cucurbits rootstocks work the same for each one. There's different compatibility issues. So you need to understand that. If you're looking to go into grafting, is you need to understand and study the rootstock that you're getting to make sure that it's going to be compatible to what you want to use it for. Now I want to give you some ideas to what goes on in the world. Okay, so if you want to look at for example, watermelons here in Asia, they use 90% of them are using the Lagenaria. Whereas if you're in Europe, only about 65–50% of them are, well, less than 30% are using the Lagenaria. So if you look at the climate conditions of both, you understand why they choose what they do. Okay, this just gives you an idea as to what goes on in the world. Is they don't all use the same type of rootstocks. And there's a reason, because of their climate conditions. What I want to show you here is the amount that goes on in the world as far as grafting. And these are in hectares, just to give you an idea. Been many trips to Korea, Japan, and Taiwan over the years. And you see how many hectares, especially in Korea, you know, and you're looking at Europe, you're looking at a lot of acreage that are all grafted plants. And what I wanted to show you here is what goes on in those countries, because they don't use any land rotation at all, basically, is you look at watermelons, 100% of their watermelons in Korea are grafted. Whereas in Europe, only 50% are grafted. And if you look at the grafted history, you look at Korea, they've been doing grafting for 30 to 40 years. If you look at Japan, 50 to 60 years they've been doing grafting. Okay, you want to know where all your rootstocks come from? From Korea and Japan. Okay, that's where all the rootstocks come from right now, except for the recent years. And so since we've been getting into grafted research, we now are one of the country's leader in the world as far as grafting goes. And it's only taken us the last 10 years to achieve that, and also to go above all these other countries because of some of the things that we've developed. And I'm going to show you those. But just to give you, this just gives you an idea as to where everything comes from. So if you look at these rootstocks, they're all related, especially the interspecifics, to a rootstock is a Shintosa type of rootstock. They all basically have, one of the parents is always a Shintosa. And they've just made selections from those for the interspecifics. Okay, the Lagenaria rootstocks that I showed you at the very first picture, that's going besides the wayside really quickly. Less and less Lagenaria is being used all over the world. And that's because there's been some problems faced with Lagenaria over the years, and so most everything now is switched to the interspecific hybrid squashes. This just shows you a picture when I was over in Korea, just to show you how this is the entire valley area, and it's all nothing but greenhouse structures like you see here. And this right here, these are just massive amounts of families that own maybe one of these. And their house is on the other end, little shack that they live in. And this is not temporary, this is permanent. And that's all they grew in there. This just shows you, this is a cantaloupe greenhouse. And this is always in cantaloupes, always. 24 months out of the year, it grows nothing but cantaloupes. Okay? And this is watermelons. Whether it's winter or summer, doesn't matter, it's always in watermelons. So, root stock disease studies. This is what we're faced with, and this is Fusarium. And this is the field that we first started many years ago in South Carolina. A grower said, you know, what's going on here? I'm losing plants left and right. And came in, we found out it was Fusarium. This is what Fusarium looks like. Most of you have experienced that. If you're at all growing any watermelons, you'll see that part of the plant will still be alive, but it won't. You'll cut a tissue like this and you see the vascular tissue. What Fusarium does is it just plugs up the water flow in the plant. This is Phytophthora. This just shows you some selections from the rootstocks that are resistant to Phytophthora. This just shows you the masses that you might have been familiar with. This shows that, and these are some of the other lines that we've tested, but this just shows you the results of Phytophthora that we've studied in our greenhouse studies. This is root knot. Okay? See this little guy here? And this is the root surface roots. That's a major concern for us. They don't have that problem overseas because they flood all their fields. Okay? And their rotation is with rice. Okay, so as soon as they flood their fields with rice, or if you have an opportunity available to flood your fields, then that takes care of that, but I doubt you do. Okay, choosing the soilless mix to grow your rootstocks. One of the biggest problems in grafting is controlling your rootstock and your scion material. Word scion just means your watermelon or your top that you want to put on. That's considered what's called a scion. Okay, so if you're going to graft something, you want to put it on top, that's the scion material. But it's getting those to grow the same stage when you go to graft. Okay, and you're going to be grafting at a very young age. Now when you look at grafting, one of the real problems is that there's tomato grafting, there's pepper grafting. Those are probably the easiest grafts to do. One of the most difficult grafts to do are watermelon grafting. Okay, they're bigger, but the problems associated with making sure that you get compatibility and you get a good matchup, and I'm going to show you that in a minute. That's the trick. That's the science. That's the art of the ability to know how to graft. But if you're going to grow rootstock material, you need to choose a soil mix that's well drained and does not have a starter in it. All the places where you go to buy your mixes all have their starter solution already in the mix that you buy. Okay? And that starter solution is generally a trade secret. And they won't tell you what's actually in the starter solution. But it generally contains nitrogen. And for a rootstock material, you don't want nitrogen. Okay, you want to grow it as slowly as possible. You need it to, as I say, free from a starter. This happens to be a Fafford mix that we use, but I insist that when we order it, we always get a mix that does not have any starter solution in it at all. Also, most of your mixes, let me back up, most of your mixes have process in them where you have perlite. Most of them come with vermiculite mixed in with the peat moss. Most of them have a wetting agent because peat moss alone, getting it to absorb moisture, you always have to have a wetting agent in the mix. So just to buy a bale of peat moss, not a good thing to do. But these simply are put in there, are processing pine bark, perlite, and vermiculite to air it up. Because you want enough oxygen in that root system. You got to have oxygen in that mix. And if it holds too much water, okay, you'll have a tendency to put conditions on it that's going to kill your root systems, and you don't want that. This just gives you what some of the seeds look like. And this is just your interspecific squash. This is your lagenaria. Looks like a bottle gourd. And then if you study these, I mean, for a gourd, when you grow a gourd, what's the one thing that you like about growing just a gourd, period? What do you like about growing gourds?
Vigorous.
What's that?
Very vigorous.
Very vigorous. Okay. What else?
Seeds typically separate from the fruit on the inside. They're a lot easier to harvest.
What's that?
The seeds separate from the meat of the fruit on the inside a lot easier.
Okay. Other ideas? Does a gourd require much water? No, you throw them outside, and I mean, if it's drier to bone, they're still growing, right? I mean, my first experience when I spilled some seed in my greenhouse and was on the floor of the greenhouse, which happens just to be a dirt floor, and I planted everything out in the field, and I just came by my greenhouse one day and noticed all this green stuff growing in my greenhouse. And it was volunteer gourds, and I mean, that had been 140 in the greenhouse during the summer and no water at all, okay, because the greenhouse cover was still on. And I opened it up and I had gourds growing everywhere, okay? So that's what's really nice about lagenaria, is it doesn't require a lot of water, okay? So what does it tell you about the root system? The root system is going deep, okay? The root system goes down, it goes deep, and it's a good scavenger for water. Okay? This one is our new one. This is Carolina Strongback. And this is a root system that we've introduced to the U.S., and it's taken about 12 years to get it to the stage we have now. But we have some seed of that in the back. And we at the USDA and Clemson University have a joint project that we released this. And it's a fantastic rootstock. Most of the wild watermelons that we've tested over the years has not been what we had hoped for. And all over the world, they've always tested wild watermelons to see if it had the potential of really doing something for us. Nobody has been able to find a rootstock that does that for them. And I'm going to share some things with you about Carolina Strongback. But it's by far much more vigorous system, and much more vigorous than either of these two rootstock materials. So I've got a packet in the back. You can distribute it, Susie, however you want to with the group. There's 1,000 seeds. Syngenta has purchased the license to Strongback. They're producing it commercially now. And so I had to get permission from them to bring that, and they just asked if you use this rootstock, that you send them pictures of how it's doing. And that's all he's requiring for that. So if you get some of this seed, make sure you take pictures of how it does for you in grafting. But if you want to auction that, Susie, that's worth a lot of money right there. It's 1,000.
I'm going to spread the love.
Spread the love. But we're excited about this rootstock, as we tested it over the years. It's really funny. We made this cross from lines of resistance to fusarium that were discovered at the USDA. And what's really nice about this rootstock is it has multiple sites of resistance to Fusarium. It also has tolerance to root knot nematode. It has phytophthora tolerance, and it also has Versilium tolerance. So it has everything that these other rootstocks don't have. Plus, it also has a more vigorous root system than either of these others. So we're really much excited about, like I said, the only drawback is that the plants that are grafted onto Carolina Strongback have a delay in the first fruit harvest of about a week to 10 days. Because it's so vigorous, the plants have a hard time switching over. The first female flowers that will appear on your plants will abort. Okay? And it's not something that you did wrong. It's just the plants still wants to grow vegetatively. The second set that comes on about a week later, it'll set them all. Okay, and what's also really nice about the rootstock is that the initial watermelon that's set, that's usually your big one, correct? Is that the one, the first one that sets, that's the one you're going to baby? Because the secondary ones out on the outside, you know, are not going to be as big. Well, with the Carolina Strongback, because of the root system, those out in the commercial, actually for watermelon production, because I know nothing about growing giant watermelons, is we have a tendency with that, with the secondary melons, to be exactly the same size as the first ones. So you get more uniform fruit set. You don't get a lag time. And it keeps on setting. What's really nice is the plants just keeps growing. And so that's caused some different concerns all over. So far it's being tested. It's the last 2 years they've had the license to test it throughout the world. And it's getting a lot of attention. A lot of watermelons in Mexico are now being grafted with Strongback, also in Australia, over in Israel, all over now. That a lot of people are very excited about this rootstock. We get calls all the time, and it's nice to see the popularity growing with this. But the U.S. is by far back on top again, so we're excited about that. Yes?
Is that a Strongback fruit right there?
That's a Strongback fruit. Yep, this is what the fruit looks like. And the seeds are going to be reddish color inside. And what's really nice about this, as far as the seed producers are, is these produce a lot of seeds. And so that makes production for this more economical. And it sets them right away. Whereas if you look at the seed company, when they talk about offering the interspecific hybrid squash seed, it's a nightmare to grow that seed. And if you ask some of the seed company people, they cringe when they go to produce this, because it doesn't set any fruit till it reaches like the 10th node. It's really out there. So the season that the crop is in to grow this rootstock is really a long, drawn out process, which means it's susceptible to all kinds of foliar diseases. It just sits in the field forever. So, and this one's not. So, I just came back from California a few weeks ago, where they just finished up a crop production of Carolina Strongback.
How did it do?
What's that?
How did it do?
Okay. Yeah. Well, it's late getting in, but it'll end up with a good crop. They'll have it for sale this year. If it's not already sold out, they might already be sold out of the seed. So it's going really fast. So they anticipated they'll be growing a lot more this next year.
So we couldn't buy more seeds, we only had to get them from you, right?
Yeah, that's probably the only one, because you don't buy you don't buy 10 to 20 pounds of seed. you only buy a handful of seed. So to get that, you're going to have to go in as a group to get. But you have 1,000 seeds back there. Now, grafting. Okay, the interspecific, you have to learn how to graft the different rootstocks and how they respond. They don't all respond the same. So if you're going to grow your rootstocks this time of year in your greenhouse, you're going to see a real difference how fast they grow. Okay, if you're going to grow in interspecifics, they will come up and open up for that first cotyledon leaf in about 5 to 7 days. Okay, they come up real fast this time of year. Okay, the coolness of your greenhouses when they're only 70, 75 degrees as you keep them now, and your groundwater is cold, which is you're going to be watering these plants with, your interspecific will respond to that rather. I mean, doesn't hurt them. They'll germinate and come right up. Okay, your lagenaria is a little slower. Okay, doesn't like the cold temperatures. Okay, so it's going to take a little bit longer to come up. Okay, and with the lagenaria, you can overwater that and kill those seeds. Okay, they like to be on the dry side. Okay, most people on the squash, they like to be watered. Okay, but these like to be on the drier side. But there's a couple of days difference there. With the Carolina Strongback, you're looking at a good 14 days here to get it started. So there's a big difference. You need to understand what you're trying to grow. Okay, and it's important when you come up to this next section, and I'll show you. This just shows you the different sizes of seed. And you got your cantaloupes, your cucumbers, and your watermelon seeds. And some of your watermelons are huge seeds because I've been working over the past several years with one of your growers in working with him on learning how to graft. Charles Bentley. Many of you know Charles, so had many conversations with him over the years, and so we've even stuck some of his plants out just to see what they were when he first said, I'm interested in growing giant watermelon. So I stole some of his seed and planted it just at the station just to see what they would do. They're enormous. We got about a 140 pounder the first time we did it, but it tastes terrible, by the way. But if you're ever growing seedless watermelons at all, we developed a watermelon transplant guide over the years. I and Jonathan Schulteis here at NC State on how to grow those. But the grafting stage I want to show you is very, very important. Now, this happens to be the interspecific squash. This is the stage that's perfect for grafting. Okay? This is ideal stage for grafting right here. As soon as those cotyledon leaves unfold, and you barely see the first true leaf coming out, that's the best stage to graft. Okay? That will change in a couple of days. Okay? And then you're way late. And then you're going to run into some problems when you graft. The one thing that you're going to need to watch out for when you graft is regrowth. This little guy right here. Whatever this rootstock is, that plant is always want to go back to what it was originally. Okay? So the process of removing this little guy, you need to keep him under check. Now we've developed a process over the years of what we call blinding the plant. And we've developed a method of applying a fatty alcohol to this growing area that actually blinds the plant and destroys the growing point on on whatever grafted rootstocks that you're growing. And we've developed a method, and I can explain that to you sometime if you're interested in that you apply this and it'll actually kill this area and blind it. Now, I want to show some things to you that are different in cucurbit grafting. This is called a hypocotyl. It's not a stem. Okay? This is not a stem. This is a hypocotyl. You need to understand because it's important when you go to graft. These really are not true cotyledon leaves. They're actually photosynthetically active leaves. Okay? You need to understand that because that's extremely important in knowing about cucurbit grafting. If you're growing tomatoes, you're talking about a stem, and you're also talking about true cotyledon leaves. If you'll notice when you have watermelon plants, those leaves always stay nice and dark and green. That's because they're manufacturing carbohydrates. They're actually true leaves. Okay? So it's very important that you maintain these and keep these healthy. Okay? Especially when you graft. If these turn yellow, you're not going to have a very successful graft. These are producing carbohydrates. And what carbohydrates do is develop the thickness of this hypocotyl and also store carbohydrates in here and also store carbohydrates down here in that stem, in that hypocotyl. And that carbohydrates, generally the plant takes carbohydrates and then converts them into sugars that develop the leaves. Okay? Or those sugars that heal the graft union. Okay? So keeping these short, stocky, not spindly at all, is extremely important to graft. Okay? That's why when I spend so much time on the soil mix that you use, make sure it doesn't have a starter. Because what happens is you'll plant these, and these will get spindly on you real quick, real fast. And you're wasting your time trying to graft those, okay? Because there's no carbohydrates in there and you'll get a poor graft. It may be a successful graft, but that graft's going to be poor and it's not going to perform like you want it to. Okay, like you expect it to. Like your neighbors do. Okay? All right? And you always get calls, well, my plants didn't look anything like my neighbors and I did exactly the same thing. I'm sure you've experienced that same thing in your operation. Okay? It's important how you grow these things. You need to understand how the rootstocks work. This is the scion material. The development of the scion material depends on the type of graft that you're doing. Okay, and I'm going to go through the various grafts with you that you can do. But it depends on the graft type that you're doing, what the stage of your scion material is. Okay, and the scion material, you want to produce it short. Okay, you don't want that stem spindly also, or that hypocotyl. Okay, you don't want that hypocotyl really tall. You want it as short as possible. Okay, and you want it as thick as possible. Okay, whereas the rootstocks, you want to have some length on this. You don't want it down here. You want it to be up here, but you don't want it up here. Okay? Because you want to have this, because when you transplant this out into the field, you don't want to bury the graft. Because if you bury the graft, you just lost the graft. Okay? Because that watermelon on top is going to reroot and it's going to say, to heck with this root system. Okay? And a lot of times I have growers that have had grafted plants and they've buried them too deep, and they say, "All my plants are dying. You must have not got me anything that had anything to do with fusarium resistance." You go out there, they're re-rooted, and the fusarium got them and took them right down. Now, powdery mildew is a big concern with your interspecific hybrid squash. And if you have a greenhouse and you're growing them in the springtime, I guarantee you will have powdery mildew problems. Okay? Sulfur is the best cure for that. You can get sulfur burners in your greenhouse that burn sulfur. Ours, they're little. You can buy them in the greenhouse industry, little sulfur burners, and ours runs for 30 minutes in the middle of the night, and the sulfur burner will take care of all powdery mildew. Okay, sulfur, very cheap, and sulfur will take care of that. But the problems you have with sulfur is you'll see this problem after you graft. And here's your powdery mildew right here. And that graft will abort. That graft will not be a very successful, very weak graft. And so you'll see that hypocotyl turn this color. Even though you don't see any powdery mildew symptoms up here, you will see it on that cotyledon leaf of the interspecific squash, but more than anything, you'll see it right here. And and sulfur takes care of that. And sulfur burners are very cheap, very easy to get a hold of, and works very well. So there are things that you're going to need for grafting. Is you're going to need clips and razor blades, and if you want to do a certain type of graft, you're going to need a bamboo toothpick. Why bamboo? It holds up, whereas the old cheap US toothpicks, once they get wet, fall apart. Okay, so bamboo doesn't. Now, here's the type of grafts that you can use to graft. There's the approach graft, there's the top insertion graft, the one cotyledon graft, and the no cotyledon graft. The no cotyledon's one that we've developed. And when I talked to you earlier about having carbohydrates build up, that's important for the no cotyledon graft. So we're going to talk to you about the approach graft. This is the approach graft. You have your rootstock material here, and you're going to make a vertical cut down below those cotyledon leaves on that hypocotyl. And here is your scion material that you want to graft, and you're going to make an uppercut below that cotyledon leaf. And then what you're going to do is you're going to slip them together like this. Okay? Then you're going to wrap it with generally a piece of aluminum foil or a rubber band or something that you can wrap this area up. And then you're going to take this and you're going to grow them about a week, and then you're going to proceed to cut this rootstock off once it starts to heal, and then you have your plant grafted right here. Any questions on that? This is done a lot in Mexico. This is the only place I've ever seen this approach graft done. Okay? And if you're just beginning to graft, this is a no-brainer. You don't have to have any special equipment to know how to do this. This works very, very effectively, only it takes twice as much of everything. Twice as much space, twice as much time, energy, but it works. You don't have to have a special room to heal them, anything like that. You can do this, okay? This is probably the most expensive way of doing it, but it works. This is the one cotyledon method. This is the most popular method throughout the entire world. Now, I like to explain the need for cotyledon leaves in this way. These two cotyledon leaves, remember, are actually photosynthetic leaves. You got to have at least one to be successful. Why? Remember, because they're actually photosynthetic leaves. They're functioning leaves. Those leaves are actually keeping this plant going. Okay? They're actually supplying carbohydrates to the root system. The root system on a plant is either growing or dying. It never stays the same. Never. Okay? It never rests. Okay? It always requires food. Okay? It may be producing fruit and absorbing stuff for the plant, but to maintain it, it requires carbohydrates. Okay? This is providing that to that root system at all times. Okay? Whether this plant is got a leaf or a stem or anything on it, it's still providing nutrients to that root system. Okay? But having both of them is a luxury. It's like us with our kidneys. We can get by with one kidney. We can't get by with no kidneys. But we can sacrifice one. And that's what they're doing with cucurbits. Because we can sacrifice one. And so what they're doing is, here's your rootstock material, here. And what you're trying to do with that razor blade, and I'll show you demonstrations of this afternoon. But what you're trying to do is remove the growing point. You got to get rid of all that meristematic tissue. Okay? Or it's going to grow back to what it was before. Okay? Or you're going to have to go in and remove it. So that's why you graft this at a very, very early stage. Okay? Also, you want to do it when it's very early, because what happens is it will develop a hollow tissue right inside there if it's too far along. And so if you graft, you'll see when it does that, you'll see a little hollow area start to show up. That graft will not work. Okay? So that's why you have to catch it at the very, very early stage. Okay, you're going to remove that one leaf. Then here's your sign material, and you're just going to take these and fold them together, and then you're going to just take the razor at a 30-degree angle and remove that. Then you're just going to put it together, and you're going to match up. You're going to match up one side here, at least one side. If the rootstock happens to be bigger than the sign material, then you want to put it on an angle so that it has a better chance of connecting. Okay? And then once you do that, you're going to put a clip on it, and I got a number of clips. I got a few hundred clips back there that you're welcome to take. You clip that on, and you want to make sure that that clip holds this cotyledon leaf up so the water doesn't get in and start to break that down. And this just shows you how it's done commercially. So here's your rootstock material. Here's your sign material. Cut at that angle. You're going to put them together and put the clip on it. See how that's pushing that up? Then you get a graft union like this. And all this is healed over and it's ready to go. Now this one is commercialized. If you got lots of money and you want to do it automatically and buy a robotic, you can have a totally complete robotic system, or you can have a semi-automatic system. The semi-automatic system is what we have. We've got one of those from Korea, and we've also modified it for some of our new grafting methods. And my semi-automatic grafting operation still requires 3 people to do it. But the grafting itself is done automatically. Okay, that does it It doesn't have a Monday morning hangover, or it doesn't have a Friday hurry and get done so I have a weekend to spend off. I can graft anytime I want to with it. This is the top insertion method. This is done a lot in China, okay, by very, very experienced growers. Now, this is by far the strongest graft there is, okay, but it's by far the most difficult graft to do. Okay, what you're going to do with this one is you're going to take your rootstock here and you're going to go in with this bamboo toothpick and you're going to go right down the center here and you're going to punch out the center just so it just barely breaks through the area. Okay, coming out the side of the hypocon. Then you're going to take your scion material and you're going to do two sides of it. So you're going to cut it down one side, then you're going to flip it over, cut it down the other, so you've got this little arrow pointed like this. Then you're going to take it and insert it in here. It goes in here and you'll feel it actually start to fit right in there. You have both cotyledons still there. Okay? But if you'll notice that the scion material, see that leaf right there? You want to basically that first leaf fully expanded if you're going to try this, rather than too small, barely coming out. Okay? I've learned this one the hard way. Because man, I tried this and I'd only get about 60% success, and I'm going back and forth. And finally, one of the visiting scientists says, "Oh, you're doing it way too early." And I tried it. Now I'm 90 to 100% successful every time. You just got to have your scion material timed to where this is actually developing that first true leaf. You can actually start to see the second true leaf coming. That's ideal. Okay. And then when you poke it through like this, remember this is just barely starting to see that first true leaf. You'll get, so this just shows you how it's done. And you can go through, it just shows you how it's done here. And then you'll insert it like this, then you'll get a graft, and then you don't have to have a clip, actually. You can use a clip just to give you a little bit extra support, but if you do it successfully like this, it'll hook on and hold itself. So what happens, you end up with a graft that looks like this. What's nice about this graft is this is the strongest graft that I know of. So if you live in a windy area, this can be around in the wind and it doesn't break apart. Whereas the one cotyledon has a tendency to want to break off in the wind. Now, I won't show you the third method because that's a very complicated method and you have to do a lot with what we did to kill out the growing point and boost up the carbohydrates. So I just wanted to show you those those three grafting types. Now the healing chamber. I always get right close to lunchtime and everybody gets real hungry. I can see them going, "Oh, I'm gonna get hungry." So I'll hurry through this. The healing chamber is by far the most important part of your grafting operation. Okay? Once you do the grafts, you've got to have a place for them to heal. You need a healing chamber that has close to 100% humidity for the first 48 hours. Okay? Because that plant, you wanna stop transpiration, basically. You don't want that plant to move water through. Okay? You want it to be perfectly happy. Okay? You want to stop all transpiration. So you need 100% humidity for the first 24 hours. Then as it begins to heal, you can start dropping that humidity. 80%, then down to 70%, and then the remainder 60 to 70% after that, till it's completely healed for a week. And it generally takes, for a watermelon graft to heal, about a week's time. Okay, we always plan on 7 days in the healing chamber. Now, healing chambers. How can I build one? These are humidifiers. You can buy these at any greenhouse store. What's really nice about it is you can build your own. These are the control units that come with the humidifier, and it has a humidity sensor built in. You put the humidity sensor inside the chamber. These humidifiers stay outside the chamber. You just have a source of water coming in, and it works the same as your toilet. It has a float, so it'll shut off and turn on when it needs more water. And this simply just blows humidity in the air and keeps this in this little tent that you made, like you see here, completely engulfed in humid air. Perfect. It's very simple to build. Okay, you don't have to be some real sophisticated operation to develop a heating chamber. But these are extremely important to make sure that you have these, because you don't want these plants to wilt. You do not want them to wilt. Okay, so any questions on these? And when the sun really gets bright outside in the spring, I mean, we can graft. If you have these, we've used them in the middle of the summer. As long as we have shade cloth like this, works just fine. People said, you know, we've had temperatures inside these chambers with watermelons as high as 140 degrees. And as long as my humidity levels are up, my grafting is 100% successful. Okay, it's that humidity controls that's very, very important. And so this just shows you with some tomatoes, and this just shows you with some melons down here what it looks like. Just plastic over the top. Very simple if you want to do your own grafting. And then the final week is out in the greenhouse, just to harden them up, stiffen them up a little bit before you plant them out into the field. We generally take a week in the greenhouse to make sure that they're ready to go. This just shows you several of the different types of grafts. And this just shows you a tray full of grafted plants that are ready. But this works very well. And from that point on, you end up with good grafting ability. These plants take off rather quickly in the field. And just remember, when you graft, that root system, which you no longer see, you have no idea how it's doing, is crucial to your operation. Okay? Has nothing to do with how pretty your plants look. Okay? Has nothing to do with how pretty they look. It's all about what that root system is doing. That's your whole key to successful in a grafting operation, is that root system. And so you need to understand that root system. And we have trays in the back, and I will show you how to graft. And as we talk this afternoon, we'll talk about what's the best method. I'll show you the meristematic growth that you need to get rid of, how to do that, okay? And show you easy ways to graft, how to hold the plants, because you can get very, very fast if you want to. I mean, I've seen people over in China and Korea, and by the way, the ladies are by far the better grafters of anybody, okay? By far. They have more dexterity in their smaller hands, better grafting than any man I've ever seen. Sorry, guys, but I can compete. I can compete with any woman grafting. Just to let you know. But over the years, that's been something I've always enjoyed. I got into grafting when we started to lose our methyl bromide label, and I thought this is the only source that we have. And right now, we have no resistance other than grafting for fusarium and root knot nematode resistance. We just do not have it. And those two diseases in the soil are constantly evolving. And what's nice about our new rootstock, it has multiple sites of resistance. And if you know anything about genetics, your site of resistance, most everything that's come about with resistance to different races has just been a single site in the genetic scheme. And that's why the resistance gets built up and moves on. Okay? And ours has multiple sites of resistance. So we have yet to see disease that takes it down. Soil-borne disease that takes it down. And it's after lunch and everybody's hungry.
We'd like, everyone that would like to go to Bibbs, we're gonna walk there. Do you have anything that, because these doors won't be locked while we're gone, don't leave anything you're afraid to lose. Okay?